Hello students, this is Dr. Faizan Mirza. I'm covering the details of DNA replication and the idea of what's the leading fragment and the lagging fragment, or you can say the leading strand and the lagging strands of DNA during replication. What are the Okazaki fragments and what is the function of various enzymes, the DNA polymerase and primarily DNA ligase. So what's happening here? We are discussing details here. So let's come to the details of this DNA replication. We know that the DNA polymerase is an enzyme that copies the DNA as it runs along the separated DNA strand. It allows complementary nucleotide to attach by hydrogen bonding. We know adenine and thymine pair up and guanine and cytosine pair up using 2 hydrogen or 3 hydrogen bonds respectively, and this is facilitated by DNA polymerase. DNA ligase, on the other hand, it is an enzyme that catalyzes the reaction of the phosphodiester bond between the two nucleotides, and it helps in making the sugar phosphate backbone. The thing that needs to be kept in mind is that the DNA, when it is there, it is being replicated. We know that the DNA is having two strands. One strand is 5' to 3', and the other is 3' to 5'. So this red fragment, this red strand, actually is the one which is 5' to 3'. So this is 5 end of this strand, and this is the 3 end prime end of this is the same strand. The complementary strand, which is blue in color shown in this diagram, will have 5' at this end and 3' at the opposite end, because we know the strands of the DNA molecule, they run in anti-parallel direction. Now when DNA polymerase keeps on working and the strand keeps opening up, this is DNA polymerase copying the strand in this direction. The new nucleotide which are added into this growing strand, in one we have a leading strand in which the strand grows in a continuous direction, whereas the other fragment, or you can say in the other DNA strand, exposed strand, we have DNA being copied in fragments called Okazaki fragments. So this is an Okazaki fragment complementary to this part, then this is an Okazaki fragment complementary to this part of the DNA strand, and this is the Okazaki fragment complementary to this part of the DNA fragment. Whereas the leading strand is a continuous strand, which is a continuous strand of this template, which is actually in this part. So why the lagging strand lags means why these gaps are left over, and why the leading strand is not lagging. The reason is this DNA polymerase enzyme. The DNA polymerase enzyme actually works by adding the nucleotides in the direction of 5' to 3'. So this is a restriction that that the DNA polymerase can only work on. So if a nucleotide is here and the new nucleotides need to be added, so it will move in the 5' to 3' direction. Then the other nucleotide will be added again in 5' to 3' direction. So as DNA polymerase keep on moving along the exposed template, it keeps adding the nucleotide in the 5' to 3' direction. You need to keep in mind that if this is the leading strand 5' to 3', it must be complementary to the end, which is 3' in this direction and 5' at its opposite end. This is how the leading strand goes on. Now the lagging strand. In lagging strand, the exposed end of the actual DNA template is 5', and this 5' when exposed, it will be complementary to an end which will be 3'. So this is where the DNA replication begins. In this case, initially the first nucleotide is placed here, so you have a first nucleotide coming in this direction at 5', then again the next goes into 5' to 3', again 5' to 3', 5' to 3'. So one fragment is laid down. Then by the moment this fragment of the DNA, this segment of the DNA gets exposed, again you have a fragment being made 5' to 3' direction, because that's the only way the DNA polymerase can function. Then the DNA unwinds further and this segment gets exposed. Now another fragment gets laid down in 5' to 3' direction, because that's the direction in which the DNA polymerase can join the fragments. And as the DNA keeps like opening up and getting unzipped, the leading strand keeps on being copied in a continuous manner, because that's the natural pathway. 5' to 3' continuity mein, it just keeps opening and it just keeps getting added up in that way. Whereas in the opposite direction, the lagging strands are there and they end up leaving the gaps in between. For example, for instance, this is the DNA strand. We know it's a complementary strand, so we know if this is the 5' and then the opposite will be 3'. If this is 3', then the opposite will be 5', and they both are anti-parallel in direction. So this again shows us that this is the same diagrammatic representation here. The complementary base pairing occurs, and the leading strand will keep on getting made complementary to this exposed end. Whereas the other one, the opposite strand, will end up having these fragments called Okazaki fragments, because every fragment is laid down in 5' to 3' direction opposite to a strand in which the 5' end was exposed at this end and not the 3'. So these Okazaki fragments are laid down, and eventually these gaps of the lagging strand are filled by DNA ligase, because DNA ligase is the enzyme that actually helps in producing joining the nucleotides using the phosphodiester bond. And when a phosphodiester bond is made, the sugar phosphate backbone keeps joining. So eventually, once the DNA replication is completed, the lagging strand will not look any more lagging, because the gaps will be filled by the DNA ligase and new nucleotide will have been placed here, and you will not have Okazaki fragments left by then. So eventually, when two DNA molecules will be made, they will be complementary to each other and they will be made by semi-conservative replication. That's it from my side. Thank you so much.